So in this chapter, we're going to talk about antibiotics. We're going to see some similarities with what we saw in chapter 9 with regard to controlling microorganisms, but that was outside the body. Antibiotics, of course, we have to take into consideration the safety factor of putting it inside the body. So most antibiotics actually are just natural metabolic byproducts that come from natural organisms that are basically going to inhibit competition in their environment. So the majority of antibiotics on the market today either come from bacteria in the soil, which include the Streptomyces and Bacillus groups, or molds such as penicillin and the cephalosporin drugs. So it's especially important that antibiotics have selective toxicity. Remember, we want it to kill them without harming us, and we're all made of the same stuff: carbohydrates, fats, proteins, nucleic acids. So we need our targets to be really specific. Most of the antibiotics on the market today are antibacterial because they have a lot more cellular structures that are different than our cells, such as the cell wall. It's a lot easier to target something that is completely different. We have very few drugs available to treat eukaryotic infections like amoeba or protozoan or fungi because they are eukaryotic, so we have too many similarities. There are even fewer antiviral drugs on the market because they have very few cellular targets to start with. If you recall, they're basically a type of nucleic acid that's wrapped up in a protein capsid and perhaps an envelope, but they don't have enzymes that we can target. They don't really have any metabolism of their own. So there are very few antiviral drugs on the market because of that. Of course, we want antibiotics to be readily available when necessary. We want them to be affordable. But very importantly, we need it to be chemically stable enough to survive, for example, the pH of the stomach if it's an oral medication, but we don't want it to be something that lingers in the body. So there's a fine line between something that lingers long enough to do the job, but can still be degraded and excreted from the body. We want drugs that can be administered easily. For example, if you're going to give a 7 to 10 day course of antibiotics, you want it to be something that the patient can take on their own at home rather than having to come in for an injection or some sort of infusion of that drug. Of course, we need it to be safe if it's going to be ingested. So when we say non-toxic, it's unfortunate, but most pharmaceutical drugs have the risk of damaging the kidneys or the liver, so we would call nephrotoxic those that have a negative impact on the kidneys. We have hepatotoxic drugs that can affect the liver. Both of those are excretion organs, so that's one reason why they're commonly a target of medications. The other problem is typically neurotoxicity. Now, of course, if it's an antibiotic that's treating a digestive system infection, such as anti-amoebic drugs or anti-helminth drugs, those, of course, usually have impacts directly on the GI. So nausea, vomiting, and other forms of GI distress are going to be common there. We want drugs that have gone through the long process of becoming safe enough to put on the market. That usually takes a couple of decades. For each medication that we have on the market, a lot of money and a lot of research went into that. So we wanted to work on more than just one organism. So having a wider range of action is both good and bad. It might be able to be used for more than one type of infection, but it also may kill the helpful bacteria in the body as well as the pathogen. So when we talk about the spectrum of action of any medication, you can see the wider the selective toxicity, then that would be considered a broad spectrum antibiotic. So things like tetracycline and erythromycin are considered broad spectrum, whereas you look at something like isoniazid, it's actually very narrow spectrum. Why? Because it works specifically on that mycolic acid of the mycobacteria. So the take-home message here is clearly no drug can work against every kind of infectious organism. So this is where choosing the proper antibiotic is really important. So there are several things that go into how we choose which antibiotic to use in a particular situation. Of course, dosage is important. If a patient doesn't take the full dosage, it's not going to be as effective. How it's administered has a lot to do with where it needs to go in the body. We have to consider what's called first pass metabolism. If you're taking something orally, a large percentage of that medication is going to get filtered out immediately by the liver before it ever gets a chance to be absorbed into the bloodstream. Intramuscular injections, usually it's somewhat of a slow release type of injection, whereas intravenous is the only way you can get a really high dose, but of course you have to continuously be putting that medication in, because once it's in the bloodstream, it will get cleared quite readily from the liver and the kidneys. So it really depends on the target of each of those medications. You have to consider how it's going to affect a fetus if it's a pregnant woman. So all those things come into play when they're deciding which is the proper antibiotic to be used. And of course, again, safety, it depends on which medication, which patient. Some patients can't tolerate certain medications because they have predisposing conditions. So if you already have a kidney disease of any kind, they would definitely avoid those kinds of medications that are nephrotoxic. Other side effects, so obviously toxicity, allergies, especially in those types of medications that have been around a long time. There's a lot of allergies to penicillin or any of the related drugs. Sulfonylamides are also something that a lot of people can't tolerate, what we call the sulfa drugs. But one of the worst side effects of antibiotics is the fact that because they do have a wide range of effects, they can actually disrupt the normal microorganisms in the body. Of course, we talked about those normal flora of the body is there for competition. So when you kill off the good guys, you can set up the situation where the bad guys can then take over. So disrupting the normal flora is a definite problem. And of course, we're going to talk a lot about resistance in antibiotics. This is becoming a bigger problem the more we use antibiotics. So we'll talk more about resistance here in a moment. So how they decide which antibiotic is most effective, sometimes they have to do what they call a CNS or a culture and sensitivity test. Technically, this is called the diffusion susceptibility test, because what happens here is the agar that's used here is called the Mueller-Hinton agar, and it basically is designed to have a very regular diffusion rate. So these medications, each of these discs, would be infused with a different antibiotic. You would inoculate the plate with a sample from the patient, and then place these discs on the plate. And then when you incubate it, the drug would then diffuse away from that disc over time. And what you would look for is what we call the zone of inhibition. So if after the incubation period you have an area where the bacteria couldn't grow, then you know that the antibiotic was inhibiting the growth of the organisms right there. So you would then measure the zone of inhibition, and don't assume that the size of the circle represents how effective the drug is. If you look down here at, for example, number 3, it's got a fairly small zone of inhibition, but it may be that whichever antibiotic is on disc number 3 has a very large molecular weight, and so maybe it didn't diffuse very far. So we have to take into consideration not just the size of the zone, but which antibiotic we're looking at. Something like number 7 or number 5, what you're seeing there's no zone of inhibition, which means the bacteria grew right up and probably right over that disc. It had no effect on that bacteria. So this is how we can determine, especially if there's a resistant strain, which antibiotic is going to be effective or not. And so the data typically looks like this. So you would take, for example, the test results for Staphylococcus aureus. When they go and measure the zones of inhibition for each of these, the results on a plate, then you would come over here and look at the drug, look at that zone. So 15 was clearly over the minimum requirement for susceptibility, which is 13 millimeters. And so the determination would then be that this organism is in fact sensitive or susceptible to bacitracin. So that would be potentially an effective drug to give to that patient if that was the infection we're trying to treat. You can see there's several other that would qualify as sensitive, but look at those numbers. It's not the larger the number, the more effective the drug. It has to do with the diffusability of those drugs. So you might think that 25 millimeters for tetracycline makes it the best drug out there, but really that's not the case. But you would have, so here you have 6 antibiotics that they might choose from for that patient. Now, if it's a patient that has immunosuppression or any kind of predisposing condition, maybe something like chloramphenicol, which would be an effective drug, may not be the best choice because that actually has the ability, it typically will cause anemia, it can cause some other side effects. So it really would look at the patient, look at the circumstances and decide which one would be most effective. You look at some of these other drugs that got tested here. We have 2 that qualify as intermediate. So they're somewhere between fully susceptible and resistant. So those might not be the first choice drugs for this situation. The worst drugs to pick would be those that qualify as resistant, right? They had 0 effect on the growth of that organism. So this test would definitely rule out some of the antibiotics that you would choose from here, because you want the most effective drug that you can that is safest for that particular circumstance. So just like we looked at when we were looking at how to control organisms outside the body, there are basically 6 different ways that we can target living cells. We're going to walk through each of these. So of course, we keep talking about how cell walls are a good target. Remember that peptidoglycan has the NAG and NAM subunits, and we have the peptide cross bridges that are formed. And so most of the common antibiotics that target cell walls basically come in here and inhibit the enzymes that are required for making that cross bridge. And we call those drugs, typically the biggest family of those drugs are called the beta-lactams, and this includes penicillin. This is the original beta-lactam drug. And so you can see here the name of the drug family is the beta-lactam. That little green square is what we call the beta-lactam ring. And so you can see that penicillin, methicillin, and all of the derivatives of those drugs have that beta-lactam or something very similar to it that would have affinity for that enzyme. And so what happens if this drug is present and that enzyme that usually comes in and cross-links the cell wall cross bridges, it would sit on that enzyme and it wouldn't come off. And so as long as the drug is there in a high enough quantity or concentration, it would irreversibly block that enzyme. And so by blocking that enzyme, the cell doesn't know any different, right? The bacteria is going to continue growing and it doesn't realize it's making faulty cell walls. And so osmosis will eventually take over and kill the cell. And so the drawback of beta-lactam drugs is that, number one, it doesn't go in and damage the cell walls that are already there. So if you have an infection that's been going on for a while, any of those bacteria that are already fully formed, it's not going to affect them at all. So again, this is part of why we need to take the full course of antibiotics, because as those bacteria replicate, it's the new ones that antibiotic can have an effect on. And so if it's an existing cell, it's going to have no effect. It'll also have no effect on any plant or animal cell that doesn't use peptidoglycan, including other bacteria. If that enzyme is the thing that the drug sticks to, then obviously it's not necessarily going to have affinity for anything else. So when we make semi-synthetic versions of drugs, so for example, we would take the beta-lactam, the original penicillin, and just change some of the side groups on that drug, then it might be more stable in places like the stomach, where it's very acidic, might help it be absorbed more readily in the body. Sometimes changing the side groups of those drugs is about making sure we stay ahead of the development of new enzymes that could then bind to the drug and just deactivate it before it can kill the bacteria. And then sometimes we're changing those side groups to then broaden the spectrum of action. Maybe it can be active against more than one type of bacteria if we change the chemistry of it just a little bit. There are other ways to target cell walls that doesn't involve that beta-lactam. Certain groups of bacteria that are gram-positive use specifically that cross-bridge uses the amino acid alanine. So if they use alanine-alanine cross-bridges, there's a certain affinity for those particular subunits when they're making their cell wall. So this is how vancomycin and cycloserine work. In some cases, the drug actually blocks the protein pump that moves those NAG and NAM subunits. Remember, those are the sugars of the cell wall. Think about where the cell wall is. It is external to the cell membrane. So cells that are building cell walls have to secrete those building blocks. And so some drugs just block those pumps. You can't make a brick wall out of brick without the bricks. And so this would be, for example, bacitracin is how that works. Isoniazid, we mentioned this is a very targeted type of antibiotic against that mycolic acid because those bacteria that use that have special enzymes and special pumps that then move that substance out. So things like isoniazid and ethambutol are used to treat tuberculosis. We can also target protein synthesis, and if you recall, translation is the second step in protein synthesis, which occurs at ribosomes. So we've talked about this earlier in the semester. If we can target prokaryotic ribosomes, we can affect their ability to make proteins. Unfortunately, we still have that problem in the mitochondria where we have the 70S type. So some of the side effects, especially on long-term antibiotics, can be things like fatigue because you're damaging the mitochondria, you're not making as much ATP. So that's sometimes a side effect associated with targeting protein synthesis. But how these drugs work is going to be either going after the small or the large subunit of ribosomes. So quick review, remember transcription, translation, these are the two steps required for protein synthesis. If it's a eukaryotic cell, of course, that first step, transcription, happens in the nucleus, but all protein synthesis requires activity at the ribosome for translation. Translation is taking that messenger RNA code and turning it into an amino acid sequence. So for example, those that we call the aminoglycosides, this is a family of antibiotics that basically has affinity for the 30S ribosome subunit, which of course again is the prokaryotic form. And when the drug binds to that portion, it basically causes that part of the ribosome to sort of bend, which means when the messenger RNA is trying to be read, you can kind of picture here if that messenger RNA is kinked, the next transfer RNA that comes in is going to miss one of those nucleotides. And so basically you're going to end up coding the wrong sequence. And so it's kind of like what happens when we have mutations in the DNA, what we call a frame shift mutation, right? These molecules work in threes. So if you skip one, everything downstream is going to be the wrong message, basically. And so if this happens, basically what's happening is the bacteria starts making gibberish, right? It's going to be putting the wrong amino acids together. There's no way that can be folded up appropriately to make the right enzyme and things like that. Another way we can target ribosomes, things like tetracycline, this family of drugs, instead of altering the shape of the ribosome, it basically has affinity for the spot on the ribosome where we start the process. And so that docking site where the transfer RNA would have to come and land, if those two transfer RNAs aren't sitting right next to each other, then a peptide bond can't form. And so essentially your amino acid sequence just stops. And so these amino acids just end up free-floating singular, and they're not going to make a sequence. Chloramphenicol, this is a drug that instead of blocking directly on the ribosome, it actually blocks the peptide bond formation. And so as long as this drug is present, anytime the messenger RNA is trying to be translated, those amino acids just never get linked together. So again, you're not going to make functional proteins if the ribosome isn't doing its job. Erythromycin falls into a family what we call the macrolides. This one actually basically blocks. It's like putting something on the railroad track, so the train can't move forward. If you can't travel down the messenger RNA molecule, then you're not going to make the full sequence of amino acids. So again, it's going to make non-functional proteins, which is going to stop the growth of that bacteria. Some antifungal medications actually target the cell membrane. Now, keep in mind, cell membranes are all made of the same types of phospholipids, but it's the other things that are embedded in that. So, for example, in our cells, we use cholesterol to stabilize the membrane during temperature shifts. In fungi, their cells don't have cholesterol. They have a molecule that's similar called ergosterol. So unfortunately, if you think about where cholesterol is processed and stored in the human body, it's the liver. So these types of medications are very hepatotoxic if they're taken systemically. So fortunately, most fungal infections are skin infections, right? Think yeast infections. They're topical. So it's fairly safe to use those antifungal medications topically. It's when you have to take them systemically that we worry about taking those medications. We worry about the safety of the liver. Some drugs are going to work against some part of the metabolic pathway. So when we have something that's called an anti-metabolic agent, think about what drives metabolism. It's usually enzymes. And so some of these drugs are going to be things like heavy metals, because if you recall, that enzyme requires a cofactor. Very oftentimes that cofactor is a heavy metal. So if you put in a heavy metal that's similar to the cofactor needed, it might outcompete the normal one, and it might cause that enzyme to be non-functional. If it's a Helminth infection, sometimes we're using these anti-metabolic drugs to just sort of paralyze the muscles that they use to attach themselves to the GI. And so they're basically just pushed out with the materials moving through there because they can no longer stay attached. Some anti-metabolic agents are going to target viruses because remember, they're going to use our metabolic pathways to copy themselves. So we'll talk about some of those examples. Some drugs in the sulfa family, so what we call the sulfonylamides, these guys are going to also work against or antagonize those enzymes that are required for metabolism. So one example of an anti-metabolic drug would be those that fall in the sulfa family, the sulfonylamides. If you look up here in the top left, the first substrate involved in making folic acid is called PABA. And so you look at the chemistry of that, and then you can look across the way. All of the sulfa drugs have that same side group. And so what's going to happen here is the first enzyme involved, this sulfonylamide, has the same affinity, so it's going to be essentially a competitive inhibitor. If the drug is there in a higher concentration than the normal substrate, it will outcompete that normal substrate, and it'll stop the process right here at the beginning. Other drugs might come in and stop the process a little bit farther down. So other drugs like trimethoprim will block the second step of that process. So keep in mind, folic acid is something that we have to ingest. It's one of our essential nutrients. Bacteria, and on the other hand, make it. And so what's unfortunate is one of the ways that bacteria have gotten around this, right, one of the forms of resistance is that if they're in your body and causing an infection, if the antibiotics work against them in this manner, all they have to do is, if they have a protein pump that they can bring folic acid in from the host, basically we are their petri dish. If folic acid is floating around in our body, they can just steal it from us instead of making it. But look what folic acid is really critical for, right? This is an absolutely necessary molecule to make DNA and RNA. So if you're a bacteria that's trying to replicate, you need folic acid. Same reason why a pregnant woman needs lots of folic acid on board during pregnancy, because those embryonic cells are replicating very quickly. So some of the antiviral drugs on the market are actually anti-metabolic agents, because if you recall, viruses are trapped in that capsid and they can't replicate unless they uncoat, or basically are released from that capsid. And sometimes it's the lysozyme enzymes that are allowing them to get out of that capsid. So if these drugs can target and neutralize those acids, then the virus can't get out and it can't replicate. So this is a common method of action for anti-flu medications. Those viruses take advantage of the one weapon that a phagocyte has to actually get out of their capsids. So this is a downside, of course, of these medications. You've just taken phagocytes out of commission, which is a really important part of our immune system. But if you keep in mind, viruses, one virus in might mean a million viruses out in one round of replication. So if we can get ahead of them in any way, those drugs can have an impact. So some drugs actually go after the nucleic acid function. And so what we call the nucleotide analogs are basically where you have a drug that looks very similar to the normal nucleotides. So for example, A, C, T's, and G's, these are the molecules at the center here. Those are the normal molecules that cells would use if they're replicating their DNA. If you look at the medications, you can see that there's a similarity. And so there's going to be an affinity for those enzymes that are helping a cell build new DNA molecules. They might, if these drugs are in there in a high enough concentration, they might choose the analog rather than the normal substance. So again, it's about competitive inhibition. So many of these nucleotide analog drugs work against viruses, and partly it's because of the nature of virus replication. It's much faster than the host cells would do. Those enzymes are much more likely to take the fake nucleotides, the analogs, rather than the real thing, which means when they're copying their DNA, they're basically making junk DNA. And so if that cell or if that virus goes on and tries to replicate itself again, it's not going to be able to, because it's basically carrying nonsensical genetic information when it moves on to the next generation. Because viruses incorporate themselves into DNA, you could also see why there's a similarity between cancer. We talked about the relationship between viruses and cancer, but keep in mind what also cancer cells do is divide and replicate faster than they should. And so that's one of those, uncontrolled cell growth is one of the characteristics of cancer. And so many of those nucleotide analog drugs that we use against viruses can also be effective against certain kinds of cancer. Because again, if the cancerous cell is making faulty DNA, then it's not going to be able to survive very long. And so that's one way we can inhibit cancer growth. Some antiviral medications, instead of targeting the nucleic acid, target their ability to attach to host cells. So if you recall that there has to be an affinity between host receptors and the glycoproteins on the surface of that virus, if the drug matches the affinity for the receptor, if it basically outcompetes the virus for landing on the proper host cell, then it can keep that virus from replicating. Now, some of these are still in research stages. There's an anti-HIV drug called Fusion, which basically has an affinity very similar to the molecule on the surface of HIV that it uses to lock into our helper T cells. So if you can keep the virus from ever locking in on its host, it won't be able to replicate itself. So most of these medications, keep in mind, especially viral medications, are really just buying time and giving the immune system a chance to go and find those viruses before they could start replicating and overwhelming the immune system. So recall that some of the side effects of taking antibiotics is disrupting the normal flora of the body. So we have lots of different organisms that live in and on the body that when you kill off one strain, you may actually give another strain a boost or a shift in power, so to speak. The balance of power is maintained very carefully by pH changes and things like that. So if you kill off one particular strain of organism, you may allow other normal flora to take over, and we call those super infections. One common example of that is when women take antibiotics, they kill off the lactobacillus in the vagina, which normally keep the vagina in a very acidic environment, right? Recall fermentation by bacteria produces acids. That acid actually keeps the normal yeast that are present in check. Yeast are inhibited by that acid. So if you kill off the bacteria, the pH of the vagina will go up, and that yeast will then be allowed to grow. It's no longer inhibited. Similarly, when we have take broad spectrum antibiotics, something like clindamycin is very commonly associated with C. diff infections. It's a broad spectrum antibiotic that kills off the good bacteria in the gut. And if C. diff is present, that's the bacteria called Clostridium difficile, and most people have at least a few of those living in their gut. If they're present and you kill off the good guys, then you're going to end up with a shift in power, and those C. diff infections will take over. And they're much harder to deal with. The immune system can't deal with the toxins that are produced. And so you end up with what's called antibiotic associated colitis. And we'll talk more about that later in the semester. So it's unfortunate that developing new antibiotics is so expensive and so time consuming that a lot of pharmaceutical companies have stopped researching and developing new antibiotics because on average, what they find is that, as soon as an antibiotic hits the market, within 10 years on average, they're going to see resistance. And so if you're the pharmaceutical company putting millions of dollars into the research associated with a drug, knowing that as soon as it hits the market, you've only got 10 years to basically make money back for the development of that drug before it becomes sort of useless, or at least less effective than it should be. So look at the history of this, right? Penicillin was discovered in 1928. It took about 12 years before they started seeing in the lab Staphylococcus strains that had developed resistance against penicillin. In fact, those initial researchers actually recommended to the scientific community that we don't release this drug to the market because clearly this is what's going to happen. So they understood resistance before the first antibiotic ever hit the market. And they were right. Within 22 years, we started seeing penicillin-resistant strains in the public, right? And so clinical cases were starting to show up. When you look at the other drugs, right, methicillin. So we started making methicillin since they saw penicillin resistance. They changed it slightly. It only took 2 years before bacteria developed resistance against that drug. It took about 15 years for erythromycin resistance to develop. But you could see that as soon as it's released, those bacteria start evolving and developing resistant genes. So some other drugs that hit the market, right? 12 years, 16 years, 3, even down to 1 year each for some of these drugs. And it's unfortunate because this has sort of created a situation where the pharmaceuticals companies really don't want to invest the time and money required to get antibiotics to market. There's sort of a push for pharmaceutical companies when they find a new drug that they just shelf it. Because if you have infections that show up to be resistant to pretty much everything available, we're going to run out of tools. So there's sort of a push to not use new antibiotics until they're absolutely necessary. Of course, pharmaceutical companies don't want to hear that because if it's sitting on a shelf, they're not making money and they're not recouping all their research and development funds. So it's really kind of a no-win situation because new medications are not being developed at a pace that we need. And most of these research has shown it's a losing battle. We're going to be chasing this evolution problem forever. And so pharmaceutical companies have come to terms with that and shifted to more research on things like vaccines or other medications that would be used chronically. Historically, there's already a problem with antibiotic development because companies look at things like this that you're only going to take once in a great while for 7 to 10 days. That's very different than something that a patient might need to take chronically for the rest of their life. They're going to make a lot more money off of that kind of medication than they will for something that is only used occasionally, and especially if within 10 years that one medication is no longer effective for the most part. So what do we mean by resistance? Well, basically this is where these organisms have developed new genes, right, that allow them to survive the exposure to that drug. And so some species are already naturally resistant, meaning they were born with the gene that allowed them to survive the presence of that antibiotic. We looked at things like isoniazid, right? It targets mycolic acid. So if you're not an organism that uses mycolic acid, those drugs are going to be completely useless against you. And so that's what we mean by being naturally resistant, right? If they're just not designed to work against your physiology, then you're already resistant. Usually when we think about developing resistance, it's because they have acquired new genes. Now, rarely what has happened is mutation, right? So organisms are constantly mutating, but to just randomly mutate a gene that allowed you to survive some chemical you're exposed to, that's fairly rare. More oftentimes what's happening is once you have a gene that's present in one species or one individual, they share it. And if you recall, those plasmids are shared with through the process of conjugation. And so very oftentimes we call plasmids R plasmids because that's sometimes the most important plasmid that gets shared is the resistance gene that the next organism takes up, and it allows them to survive exposure to that antibiotic. So keep in mind, we don't make them resistant. Their genes make them resistant. What we do is facilitate that process, right? So you can picture sort of if instead of a petri dish, this were a patient's body. Chances are, amongst all the organisms in that infection, there's probably one or two mutants in there that maybe have that resistance gene. If you expose those organisms to the antibiotic, you're going to kill off all the sensitive ones and basically allow those resistant ones to survive. So if you don't take the full course of antibiotics, what's going to happen is your immune system will have to be taking care of all those other organisms rather than maybe focusing on the few that are left over that are resistant to the drug. You're hoping that your immune system can cover those guys, right? So your immune system has tools that the drug does not. And so what happens is we basically select for those organisms. Every time we expose a population of bacteria to an antibiotic, we're basically selecting for the resistant strains. And so when this happens over and over and over between different patients and over time, we're basically selecting for those that can survive in the presence of that antibiotic. So what kind of genes are they developing? Well, very commonly they're going to be enzymes. And so remember your beta-lactam drugs, like penicillin, those organisms that are penicillin resistant, this is usually the most common form of resistance. They have developed enzymes called beta-lactamase enzymes. And that beta-lactamase enzyme basically goes into that penicillin, that beta-lactam ring, and cuts it. So you can see here on the right, if this is what penicillin looks like when it's been exposed to an organism that has this beta-lactamase enzyme, then clearly that penicillin no longer has affinity for the enzyme that was trying to block that cell wall. And so that's all they needed. They just needed the one gene that makes the one protein, which is this enzyme, that totally deactivates the drug. So as long as this drug is present, it's not going to have any impact on the cell. Another way that they might become resistant is if they develop enzymes that allow them to abandon whatever pathway that the drug was blocking. So for example, we looked at that folic acid synthesis pathway. If you are a bacteria that is resistant to those sulfonylamides, it's probably because you have a protein pump that can just pump folic acid into the host cell or into the bacteria cell from the host. And so this may or may not go on forever, right? As long as the drug is present and keeping them from making their own folic acid, they'll just take it from the host. Once the drug is gone, because the 7 to 10 day course is over, then the bacteria will probably just shift back to doing its normal thing. As long as nobody's blocking their enzymes, they'll do the most efficient thing, which for them is to make folic acid. And so again, they can just temporarily abandon what they were doing if the drug is keeping them from doing that thing. A very common mechanism of resistance would be when they develop a protein pump, which very oftentimes we refer to as resistance pumps. And so it's just what it sounds like. When the drug diffuses into the bacterial cell, they have a protein pump that has affinity for that drug, and it'll literally just spit it right back out before it can affect the cell. So, for example, if it's a drug that works on ribosomes, if the cell is constantly pumping it out before it can go and bind to the ribosome and do any damage, then the organism is going to survive, and it has now resistance to that drug. What happens when we have pumps develop is very oftentimes the shape of the pump can be such that more than one drug that are similar in structure can cross through that same pump. And so we call this cross resistance, right? If they're drugs that are all in the same family, remember when we make semi-synthetic drugs, we're really just changing one little side group. And so if the drug looks fairly similar otherwise, that same pump can pump more than one kind of drug in that family. If they can pump more than 3 drugs through that same pump, we're going to call those multi-drug resistant. So you may have seen this MDR infections. You'll see XDR. I've seen this both extensively or extremely drug resistant. And this typically involves drugs that are in more than one family. So if it can move more than just the single group, so what we talk about second line, third line, every time you change the chemistry of that drug, it becomes a new generation of the drug, so to speak. So if it can move more than one generation of drug, it's considered extensively drug resistant. We now, unfortunately, have what we call pan-resistant strains, meaning we have yet to find a drug that's effective against those infections. And so these are the ones we really worry about. Once you have no effective medications on the market, what is that patient to do, right? The more resistance that develops, the more you have to rely on your own immune system to get past these infections. And so this is really the consequence to society of people misusing antibiotics, is the fact that we're eventually going to have no effective medications on the market. So when people go to their doctor with the flu and they insist on getting antibiotics, when antibiotics will clearly not affect a virus if it's designed to affect a bacteria, all we're doing is developing more resistant strains. And so we call these resistant strains superbugs, right? We said super infections were when your own microorganisms get sort of a balance and power shift. But a superbug is one that has one of these resistance genes. And so we're seeing more and more superbugs in places where, of course, where a lot of antibiotics are used. There's a statistic out there that, at any point in time, if you walk into a daycare, 80% of the kids in that daycare are on an antibiotic. And so, of course, daycares, they're all sharing toys, sharing spit, everything is being transferred quite readily. In hospitals, of course, we have staff that are moving from patient to patient. And so there's a constant exposure, basically, between sensitive cells that are being passed around. Now, historically, when we thought about resistance, they were all hospital acquired, meaning most of those strains were coming from the clinical setting. Now, unfortunately, we're seeing superbugs that are being passed around in the community. Some of the first strains that we considered community-acquired superbugs were things that were being passed around. For example, MRSA, methicillin-resistant Staph aureus infections in tattoo parlors and places like that. So it's become much more widespread than just in hospitals. And so again, why it's more important in those places where you have lots of antibiotic use and you have lots of crowding, patients close together that may have different kinds of bacteria that are sharing these genes. It's any place that those guys come in contact with each other, because conjugation is more readily managed when those bacteria are in the same place at the same time. So some examples, we have MRSA, VRSA. Now we have Staphylococcus that's resistant against vancomycin. Multiple drug resistant tuberculosis. This has become a real problem we think of tuberculosis as sort of an ancient disease, but it's becoming a bigger problem globally because of drug resistance. So we'll talk about that when we get to the respiratory system. Even C. diff infections, we're seeing some antibiotic resistance, which is becoming really problematic in the clinical setting. We'll talk about some of these other examples, CRE and CRKP. So what can we do about it? If we want to slow down the development of resistance, number one, we need patients to take the full course of antibiotics. So it's unfortunate people take home a 7 to 10-day course. Maybe on day 3 they start feeling better, so they just stop and they think, well, I'll just keep these other antibiotics in case I feel bad somewhere in the future. Well, all they've really done is narrow down the population of bacteria in their body to those that are resistant. And so when they start growing, if they were to go back and start using that antibiotic again, it's going to be useless. And so we're just encouraging the evolution of these organisms by not keeping the full dose and exposure on those populations. Another way that we can try to get ahead of this resistance problem is by combining drugs. So we saw that briefly when we were talking about the folic acid pathway, synthesis pathway. So if you attack 2 different enzymes, it's much less likely that that organism can develop 2 different kinds of resistance at the same time. And so when we combine drugs, not only do we minimize the toxicity. So for example, sometimes drugs, each one is very toxic, but if you're using them in combination, you can drop down the dose of each one, and so it's a little bit less toxic sometimes. But what we really hope for is that they enhance each other. So what you want is synergism. You want the zone of inhibition here. You can see where the 2 drugs were both diffusing into the same space. It had a more effective inhibition. So the zone of inhibition there in the green is much bigger than either of those drugs were alone. What you don't want is a drug that's going to be antagonistic to another. So this is unfortunate where some patients take their, take the circumstances into their own hands and start mixing medications that they might have on hand. They might actually be working against each other. For example, if you have one drug that's stopping protein synthesis at the ribosome and the other drug is supposed to be blocking cell wall synthesis, they're not going to be making cell walls if you're shutting down the ribosomes. So those 2 would not be an effective combination. They would actually be working against each other. So really the main thing we've been doing to date is just changing the drugs consistently. So if you just change the side groups on those original molecules, then maybe it won't have affinity for the enzyme that the bacteria has developed. And so this is where we get the names 2nd generation, 3rd generation. Some of these classes of antibiotics, we're up to the 7th generation. We just keep changing the drug, hoping that the bacteria don't figure it out. But clearly we're losing this race. So it's unfortunate. So that's why we have to approach it from different methods. This is clearly not a sustainable method of stopping the resistance. We talked about quorum quenching, right? If you have an infection that relies on that quorum sensing communication, if you can find a drug that just keeps them from communicating, you can maybe keep those organisms from producing the toxins. This is one of those that C. diff is at the top of the list of research for this because really the damage from C. diff comes from the toxins that are produced once they get into that quorum sensing stage. And because C. diff infections were driven by antibiotics in the first place, one of the worst things you can do for a patient that's got C. diff is just give them more antibiotics. They're already maybe had been exposed to some toxic antibiotics. Their kidneys and their liver are already potentially damaged. So the last thing you want to do is give more antibiotics. So this is one of the areas that we're trying to deal with C. diff infections. Some other important examples we've seen recently, this is about a decade ago now, but carbapenem-resistant Enterobacteriaceae. These were the cases we saw. There were over 150 cases tied to a single endoscope that had been not properly maintained and disinfected between patients. So you can see why if you're inoculating this bacteria directly into the GI that was resistant to antibiotics, those patients, many of them died because of that exposure. In India, we have resistant strains. So not only multi-drug resistant, we now have some extensively multi-drug resistant, extensively drug resistant strains of tuberculosis forming in places like India, where tuberculosis is much more common than it is here. And partly because over there we talked about isoniazid being a very effective drug against that organism. But it's because in that part of the world, you can actually buy isoniazid on the street and it's marketed as a lung vitamin. So basically, people are taking it prophylactically, and that has driven the resistance. So isoniazid actually went from being the most effective drug to almost like 80 or 90% resistant in that part of the world within 10 years. And so this is really unfortunate because this is a difficult disease to deal with in the first place. And now one of the two most effective drugs, there's a lot of resistance against it. In some cases, it's about black market drugs. And so when malaria, this is sort of the classic case where this problem has come to light. When you have counterfeit drugs on the market, what happens is they typically take the real drugs and they dilute them down to the level that if they were tested, they would test positive for having the active ingredient, but they're so diluted and repackaged that if they're taking that medication as indicated, the dose is way too low to be therapeutic. So all we're really doing is selecting for those organisms that are resistant to that drug. And it's really unfortunate because this is a treatable infection. If you're an infant, however, that medication is absolutely necessary for survival. So what's unfortunate is you have maybe parents who are buying the drugs on the black market thinking that they're treating their infant. And by the time they realize that the medication's not working, that disease has become fatal. Adults can survive malaria with or without the drug. It's not pleasant, but it is survivable unless you're an infant, and so it's really hard on their physiology. And so you might think, well, why do we care about malaria? It's in other parts of the world. It's not here. But realize that pharmaceutical companies are typically in the U.S. and Europe. And so when you have counterfeit drugs on the market, when they finally get found out, very oftentimes those counterfeit drugs are using packaging that look like the real thing. And so in order to get ahead of the black market, they have to repackage, they have to redesign their packaging, redesign their labeling and things like that to get ahead of those black market drugs. And that is expensive. And so that financial burden is one of the reasons why drugs are so expensive. The U.S. and I think it's maybe Sweden, I think, are the two countries in the world where it's legal to advertise prescription drugs on the television. So one of the ways that pharmaceutical companies make back their research and development funds are by advertising, right? So if you've heard about a drug on the TV when you go see your physician, you're going to ask for that drug, and that has a huge impact financially for those companies. And so it does have an impact on us in the financial realm. And of course, again, it's driving resistance. So those infection strains they're out there, and everyone is susceptible. So what can we do about it? It's clear that we're never going to get ahead of the evolutionary race. Those bacteria will continue to change as we expose them to new antibiotics. And so the best thing we can do is restrict the use of antibiotics. This legislation, PEMTA, is something that's been proposed every year, in fact, since 1999 here in the U.S. to try to restrict the use of antibiotics in agriculture. We know millions of pounds of antibiotics are used prophylactically in agriculture. So, for example, if you're going to raise livestock in a crowded condition, they're very likely going to get sick. And so by giving them antibiotics prophylactically, it not only keeps them from getting sick, but it's actually shown to have increase their growth rates. And so some theories propose that maybe that's because if you're in a crowded condition, your immune system is going to have to work really hard to keep you from getting sick. And so by sort of distracting that by giving antibiotics, it gives those animals all the energy is then shifted into growth rather than immune defenses. And so that's one reason why it's used. And so this legislation actually would restrict the use of not all antibiotics, but only actually the 8 classes of antibiotics that are important for human health, what we call medically important antibiotics. And so other countries have figured this out. Most of Europe has already got legislation. They've had it since the early 2000s. The FDA in 1977 figured out there was a link between antibiotic use in livestock and the resistant strains that they were seeing in human beings. So this is nothing new. We've known for decades that this was a problem. In fact, at the time when they proposed this initial legislation in 1999, there were already 500 scientific studies showing that the livestock industry was the source of most of our antibiotic resistant strains that were hurting human beings. And so trying to get this legislation passed has been very difficult because we have a very powerful lobby from the industry to keep this type of legislation from hitting the books, so to speak. So because it's been very problematic to get that legislation passed, there's sort of a new approach to just reclassify how antibiotics are regulated. And so if they got changed to what's called a societal drug, just what it sounds like, those who are taking antibiotics have potentially an impact on people around them. So if they're taking them inappropriately, and they develop an antibiotic resistant strain that then gets spread to other people, that has an impact. And so this actually may be more effective than trying to restrict the use altogether, because really most pharmaceutical drugs now are really only affecting the individual that they are prescribed to. But because the agricultural industry has a very powerful lobby, and there's so many legal loopholes in the way that we use antibiotics today. So for example, antibiotics are listed as a food additive. It's actually considered a safe ingredient by the FDA. And so when you buy something at the grocery store that came from an animal that was fed antibiotics, most people have this misconception that you should be afraid of the antibiotic in your food. It's not the antibiotic you should be afraid of. It's the fact that the animal was raised with antibiotics, and any bacterial infection that they have is very likely to be antibiotic resistant. So if you have salmonella contaminated meat, if you don't follow the packaging where it says to handle it properly and cook it fully before you eat it, you're potentially exposing yourself to antibiotic resistant strains of bacteria. It is not illegal for them to sell you bacteria contaminated chicken. Why? Because that label told you to cook it. If you didn't cook it, that's on you, not on them. And so if we could make pathogens, if we could regulate the meat industry to consider any pathogen on that meat as an adulterant, then it would be illegal for them to sell bacteria-laden chicken. But because it's not, that's a problem. And so really the antibiotics that this legislation we were talking about, it only would restrict those antibiotics that we consider medically important. There's good evidence that shows most of the antibiotic resistant strains we see in humans can actually be tied to the livestock industry. And so that connection should be enough evidence that there's a problem. But again, other countries have figured this out, they've restricted it, and they have seen less antibiotic resistance in some of those organisms that they previously were seeing, just like us. So it's clearly a problem. If you're going into healthcare, it should be important to you because you're going to be exposed potentially to those organisms. There's some statistics, usually what I saw, the latest statistic is 60% of hospital infections are antibiotic resistant. So if you are going into healthcare, you are on the front lines of exposure to those organisms that you may or may not be able to treat with antibiotics. So clearly it's a big problem. All right, that's it.